Hello everyone, this lesson is going to be on the cell cycle and mitosis. So what we're taking a look at here is kind of a pie chart, which is showing the various different names for the phases of the cell cycle, different stages of the cell cycle. And we'll also talk about the different events that are taking place that do go along with each one of these phases. Overall, what is going on with the cell cycle is we're starting with one single cell, and by the end of the cell cycle, we now have two cells. The start of the cycle we usually consider to be this point right here. So it's here that we have one single cell, perhaps one that looks like this. And then as it does go through this entire cell cycle, it reaches that point again. Now instead of one, what we end up with are two cells. The major part of the cell cycle is referred to as interphase. Interphase is the phase of the cell cycle where the cell is not dividing and it's for the most part carrying out most of its normal daily activities. On average, the cell will spend about 90% of its time in interphase. The remainder of its cell division then would be about 10% of the cell cycle. Picture that we're taking a look at here, it does have a direction, and the direction is clockwise. So if we take a look at the arrows that they show here at various different locations, that's the pattern, that's the sequence that we're going to follow. So at the start, we're going to have G1, followed by the S phase, G2, and then we go into the various different substages of cell division. When a cell is initially formed, it does go through this G1, which for the most part is a growth phase. So during this growth phase, if you think that a cell did just divide, so maybe that cell needs to grow in terms of its overall size. So we're going to see that taking place, and then it is again just going to be carrying out its normal daily activities. There are some cells that will never divide, and they then kind of exit out of this cell cycle and they go into what is referred to as G0. So G0 is the period of time where the cell is carrying out its normal activities and it will continue to do that and will never divide again. That is G0. For cells that do go on and divide, they will progress through the remainder of the cell cycle. So what we still have in interphase is a cell that has kind of made the decision that it's going to go on and divide, and then it progresses from G1 into S. So the S in this case does stand for synthesis. And what we're really talking about is the synthesis of DNA or the replication of the DNA, and that is the chromosomes. So even though this is not when the chromosomes are dividing, that's not when the cell is dividing, this is already in preparation for cell division, where we're going to take each one of the chromosomes, 46 chromosomes if we're talking about humans, and we're going to make another copy of it. We're going to duplicate those chromosomes. All of this is taking place during interphase. And remember that during interphase, chromosomes are in the form of chromatin. So when it is in the form of chromatin, it's long and stringy. It's on kind of that beads on the string kind of formation. You can't see the chromosomes because they're not really in condensed form. So you would not actually see the chromosomes looking like this, but this is the time that we do have the replication or the duplication of the chromosomes. Multiple different what we call restriction points or checkpoints along the way. So we would have one right here, for example. I'll put it as R for a restriction point. So it's kind of at this stage that that cell, in consultation with other cells in the surrounding area, is going to come to the decision as to whether it needs to go ahead and divide. So if that decision is made, then it is going to go from G1, it's going to go into S, and it's going to then start to get ready for the division of the cell. After the chromosomes have been duplicated, then it goes into a second G, which is G2. So this once again is growth, only now it's not just growth that's taking place, it's growth and preparation for cell division. So whereas G1, we say growth, growth and interphase and the normal activities. G2 is more specifically growth and also growth and preparation for the cell division. And again, at various different points along the way here, there would be checkpoints, there would be restriction points, just to kind of make sure that everything is progressing as it should, and there are no problems with the replication or the growth prior to entering into the actual cell division phase. So now that we're at this point here, the cell is ready to go ahead and go out of interphase and into cell division. Cell division, we divide up into two different components. The first one is mitosis, and mitosis refers specifically to the division of the chromosomes. So I already did show that over in S, what we're going to be doing is taking each one of those chromosomes in the form of the chromatid, duplicating it. Now when it does go into mitosis, this is now going to become visible. And what's going to take place throughout the various different stages of mitosis that we'll get to shortly, prophase, metaphase, anaphase, and telophase, is we do want to take these two sister chromatids, pull them apart, and eventually divvy them up between the two new cells that are eventually going to form. So all of that, very, very quickly, is the division of the chromosomes, and that is specifically what the term mitosis is referring to. There are many other things inside of the cell that also need to be divided up evenly. All of the internal organelles, the cytosol, you want to kind of make sure that each of the two newly formed cells get about the same amount of each of these. So not only the chromosomes, but the other cellular materials. So when we talk about the division of everything else except for the chromosomes, that is what is referred to as cytokinesis. So I'll just put that down as division of the cytoplasm. So cytoplasm, we're talking about the fluidy portion, the cytosol, and also all of the internal organelles that are in the cytosol as well. You'll also notice, or hopefully notice, that in this diagram, we do have a little bit of overlap between mitosis and cytokinesis. So we still have the chromosomes that are being pulled apart and split up between the two newly forming cells at the same time that we're starting to have cytokinesis taking place. So there is some overlap. Usually we talk about the last phase of mitosis, telophase, overlapping with the division of the cytoplasm, overlapping with cytokinesis. So now, whereas at the beginning of cell division, we had one single cell in prophase, as we eventually progress through the end of telophase and cytokinesis, now we end up with two cells. Each one of these cells now would then continue on with the cell cycle, enter into G1, and if they needed to divide, eventually progress through S, G2, and so on. So let's take a closer look at the actual process of mitosis. So in this picture here, we do see that we have these four different phases: prophase, metaphase, anaphase, and telophase. And just like with the cell cycle, where you do need to know the order, so the order is G1, going to S, going to G2, going to mitosis and cytokinesis. That is the order, that is the sequence that you need to be aware of. You also need to be aware of the order of mitosis as well. So yes, it is prophase, then metaphase, then anaphase, then telophase, and of course that overlap with the cytokinesis. So let's take a look at the events that are going on with this process of mitosis, and overall, what does mitosis accomplish? So the point of mitosis is to produce two cells, and these cells are genetically identical cells referred to as the daughter cells. So we start with one and we get two. Before the cell division does take place, we do need to duplicate the chromosomes. Again, that is during S of the cell cycle, and we do that only once. So we duplicate the chromosomes once, and we split up the chromosomes once. So if we're starting with a diploid cell that has 2 copies of each chromosome, it's going to conserve that chromosome number, which means keep it the same. So we start with one cell, which is diploid. We end up with 2 cells, and both of those are identical to each other, and they are diploid as well. So what this is for is, well, growth that might take place during embryonic development, fetal development, and even once the baby is born, repair, replacement of cells, and for some organisms, asexual means without sex, and that is their form of reproduction. The picture that we're taking a look at here is a microscope image, which is showing some plant cells. And what we do see in this one here is a whole bunch of cells where we can clearly make out the nucleus. So all of these here, we can see the nuclear membrane. But if you take a look at the inside, we really can't see the chromosomes. And that's because all of these, they do have their chromosomes, once again, in the form of chromatin, and it's in the uncondensed form, and these are the cells that are then in interphase. This is what a cell looks like when it is in interphase. And once again, I do want to stress that it's not that these cells aren't doing anything, they're doing everything. They're just not dividing. If we take a look at some of the other ones that look different, what we can see is, well, now there are some thicker lines that are visible. So these ones that I'm circling here, they are ones that have entered into mitosis, and they're at various different stages of mitosis. So by taking a look at some of these, we can see that this one here is probably in one of the phases that is referred to as anaphase. This one here is probably anaphase as well. And the other ones, most of them, they look like they're probably in the first phase, which is prophase. So what are the details of each one of these phases? What do they look like? And what do you actually need to know? So some really nice pictures here, quite simplified pictures. So with the pictures that we're taking a look at, these are diploid cells, but they are not cells that have 45 chromosomes. In fact, each of these here, if we count them up, they only have 4 chromosomes. 4 chromosomes or 2 pairs of chromosomes. So what are the pairs? Well, we have a longer dark blue one, and we have a shorter dark blue one. So those two would be the homologous chromosomes. And the other ones that we have in the middle here, the 2 shorter ones, one darker colored one and one lighter colored one, those would be the other chromosome pair, the other homologous chromosomes. So the first picture that we are taking a look at, this is a cell where the chromosomes have already been duplicated. So if we kind of zoom in on this one here, we would have started with one single chromosome, and then during S of the cell cycle, this is what happened. Other one is made. They're kind of sandwiched together. So in this image here, it's kind of difficult to make out the fact that yes, it has been duplicated, but this is a duplicated and partially at least condensed chromosome. Sometimes they will show them as kind of an X-shaped structure like this, but we don't see it in this particular image that we're taking a look at here. So in this image, what we do see is that the nuclear membrane is still here. The chromosomes are at least starting to condense. We do have some structures that are also shown here. These are called the centromeres, and within each centromere, eventually there will be 2 what are called centrioles. And these centrioles give rise to these fibers that are sticking out. And these fibers that are sticking out there are what are going to move the chromosomes around, and they are called the spindle fibers. The spindle fibers. So for our first cell that we do have here, because we do still have the nuclear membrane hasn't disappeared, because we don't really have the centrioles or the centromeres or the spindle fibers that are all that fully developed. And again, I said here, it's kind of questionable as to whether they really are condensed. So this one here, what I'm going to do is I'm going to call this one an interphase cell. So with interphase, the nuclear membrane would still be visible. the centromeres and centrioles would not be fully developed, and spindle fibers would not really be forming yet. And in reality, if this were interphase, we wouldn't be able to see the chromosomes, but they would have been duplicated at this point. So interphase is not one of the phases of mitosis. All of the other ones are phases of mitosis. So the first one here is prophase. And there are a few different events that you should know about prophase. So if we compare interphase to prophase, what can we actually pick out? Well, first of all, the nuclear membrane disappears. Okay, so it's eventually going to disappear. Second thing that we see is for sure here, the chromosomes condense. And finally, we can see that the spindle fibers are forming. So these three events are really the big ones for you to remember that are taking place in prophase. Now, if we take a look at the next stage, and one of these does just naturally flow into the next one, the next one is called metaphase. And during metaphase, what we can see is that the chromosomes that were previously just kind of randomly scattered throughout the cell, now they're going to line up all along the middle. So this middle is referred to as either the metaphase plate or the equatorial plate. So equatorial plate, if you kind of think about the Earth, about the globe, the North Pole, the South Pole, and the equator, we're going to talk about the location of the cell using that same language. So along the middle of this metaphase cell, we have the equator and we have two poles. We usually don't refer to these ones as the North and the South Pole, but this is one pole and this is the other pole. And notice that that is where we have the centrosomes, and within each centrosome, the two centromeres, one at this pole, one at the other pole. Radiating out from those centromeres is where we find the spindle fibers. It is the spindle fibers that are going to attach to the middle of each chromosome, which is the centromere. And it's these spindle fibers that are going to move the chromosomes around. So they are responsible for moving the chromosomes that are randomly scattered throughout the cell and lining them up along the middle, along the metaphase or the equatorial plate. And that's really the only big thing for you to remember with metaphase. The chromosomes line up. Now, I should point out that here it does show them lining up horizontally, but it doesn't have to be horizontally. They could line up in any orientation. They could be lined up on an angle like this. They could be lined up vertically, going up and down. They are lined up along in one particular direction, and at the opposite ends, again, would be those poles. So now we have prophase, metaphase, and again, this is the order that they do go through. The next one that we have is anaphase. So this one is typically the shortest one when we talk about the different stages of the cell cycle, different stages of mitosis. Now what we have is the chromosomes that were lined up. The spindle fibers are going to be attaching to the chromatids, and they pull the chromatids in opposite directions. As soon as they're pulled apart, though, they're no longer called chromatids. They are now chromosomes on their own. So half of the chromosomes go toward one pole, the other half of the chromosomes go toward the other pole, and that's really the only event that you need to know with anaphase. Again, they were chromatids attached, forming one single chromosome, a duplicated chromosome, but when they are pulled apart, they are called chromosomes once again. And then we're on to the final phase of mitosis, and this is where we would also have cytokinesis occurring at the same time. So this one here is Tito Phase. And during telophase, we're kind of reversing some of the events that took place over in prophase. So what happened in prophase? The nuclear membrane disappeared. What's going to happen in telophase? The nuclear membrane is going to reform. We have the chromosomes that are now at the poles. We see that. So with anaphase, they're moving toward the pole. When we get to telophase, well, now we say that they are at the pole. During prophase, they condensed and they became visible. During telophase, they're going to decondense. So think of it in some ways as a reversal of the events that we did see taking place in prophase. So all of these again would then be the phases of mitosis. Interphase itself is not one of the phases of mitosis. Mitosis, once again, is referring specifically to the division of the chromosomes, and that's what has taken place. The last image that we see here, we do see something else going on, and that's with the cell membrane. So in this case, we see the cell membrane that's pinching in on either side, and this goes along with divvying up everything else inside of the cell. So even though we do have the final stage, telophase, maybe even anaphase as well, at the same time, what is also going on here is the cytokinesis. And that then completes our cell division. We started with one cell, now we have two cells. Those two cells are going to be identical to each other. Just one other picture we'll take a look at here. These are plant cells we're taking a look at. More or less the same process. So the different stages, once again, that we do have. This is our interphase cell where we can't see the individual chromosomes. Nuclear membrane is still there. We don't see the spindle fibers. This is prophase. Nuclear membrane disappears. Chromosomes condense and become visible. Spindle fibers form and start to move the chromosomes around in the cell. This is our metaphase. Chromosomes line up along the metaphase or the equatorial plate. This is anaphase. They are pulled apart. And this is prophase. Chromosomes reach the poles, they decondense, and the nuclear membrane is once again going to be forming. In this picture here, we don't see the pinching in because plant cells have a rigid cell wall around the outside. So instead, what we have forming in the middle is what is referred to as a cell plate, and that will eventually form the membrane and the cell wall between these two newly forming cells. Same thing with the images inside the nucleus. We're taking a look at the red and purple stained chromosomes. We don't see any on this one here. Now we can sort of make out some of the details of the chromosomes. They're spreading a little bit more around in the cell because there's no longer a nuclear membrane. Chromosomes are lining up. They're being pulled apart. They're decondensing, they've reached the poles, and again, we can see the cell plate that's forming in between to eventually give us two new plant cells.